Well, jumping straight in, we've probably seen already at this conference several graphs that show that atmospheric carbon dioxide has increased and continues to increase. And in fact, last year we saw the largest step increase in CO2. And part of my research group's work is really to look at how aphids respond to elevated carbon dioxide concentrations. So aphids, sometimes called green fly, black fly, white fly even, transmit about 50% of plant viruses that are vectored by insects. So they're an economic pest in their own right. But the other thing about this group is that they seem to be the group that most consistently benefits from elevated CO2. So various reviews and meta-analyses tend to demonstrate that aphids benefit from elevated CO2. Not always the case, but it seems to be the group that you can most consistently say this about. It's also a group of insects that has a sort of dizzying array of interactions with antagonists and mutualists. And that's really what I want to talk about today, really to not only address how aphids are affected by elevated CO2, but how some of the interactions that aphids have with other organisms are shaped by these changes in our climate. So really there are three parts to this talk. First of all, I'm going to talk about how elevated CO2 and temperature affect lucerne aphids through changes in amino acids. Secondly, I'm going to say how this, switching to a different system in raspberry actually, I'm going to talk about how CO2 affects interactions with natural enemies of aphids. And then turning to a completely different interaction in mutualism, I'm going to talk about how elevated CO2 affects interactions with mutualistic ants based on changes in host plant quality. Okay, so just a quick word about environmental change and plant quality. Generally speaking, I guess most of this comes from crop plants, but elevated CO2 makes plants less nutritious in terms of nitrogen at least, because it drives plants to have a higher carbon to nitrogen ratio. Again, it's not always the case, but this seems to be the most consistent trend in terms of carbon nitrogen. Legumes, which can undertake biological nitrogen fixation, differ from this because of their ability to fix nitrogen. And because CO2 and temperature are likely to increase in tandem, we've got to sort of ask whether these two factors are going to drive nitrogen fixation differently. And there's good reason for thinking that elevated CO2 increases nodulation because as plants get bigger, they have a bigger nitrogen demand. So essentially, they need to undertake this symbiosis and obtain more nitrogen. But at the same time, nitrogen-fixing bacteria are not terribly good at coping with higher temperatures. So you can see how these two factors might have opposing effects. And this is what we actually see. This is work by James Riles, but actually there are several other studies that show very similar patterns for other legumes. This is in lucerne. So what we can see is under ambient ambient temperature of 26 degrees, this is Australia, remember, we can see that in the grey bar, nodulation's increasing under elevated CO2. But as soon as you increase that temperature to 30 degrees, just plus 4 degrees, then that effect is pulled right back down, and the plants aren't nodulating nearly as much as under ambient temperature elevated CO2. So the first aspect of the work I wanted to talk about is how CO2 and temperature affect lucerne aphids. And as I say, this is work that James did, working on P. aphid, and he was interested in how these two factors could affect both the P. aphid's performance, but what were the underlying plant-mediated mechanisms? And he focused on phloem amino acids, which are kind of the most important nutrient, really, for aphids. So he set up his experiment. I won't go into the details of it, but he worked on 5 very disparate lucerne cultivars and measured a whole raft of insect performance parameters. So this is what he found. So again, this is quite similar to the nodulation graph. We've got the ambient temperature here. And for all of these performance parameters, you can see that in the green bar, which is the elevated CO2 treatment, you can see that the performance in each case is increasing. But as soon as you throw elevated temperature into the mix, in most cases, these performance parameters are decreasing. So it's kind of mirroring the effect that you see in patterns of nodulation under elevated temperature and elevated CO2. And what James found is actually across all these 5 cultivars, which are very different in their morphology and their origin, their parentage, is it was always these 5 amino acids here that seem to underpin the effects of temperature and CO2 on aphid performance. So when CO2 basically caused this group of amino acids to increase, it was actually temperature that brought those concentrations down. So the other amino acids, these were not really underpinning any effects, and these ones were underpinning cultivar effects, but that's a story for a different day. And this just sort of underpins this tight relationship between the concentration of these 5 amino acids and the performance of aphids. In this case, it's just aphid fecundity. Okay, moving on to the next part of my talk, which is to do with the interactions aphids have with natural enemies under elevated CO2. So animals that live in groups, and aphids are amongst this, often use alarm signaling to warn each other of impending danger. And in the 1970s, it was discovered that aphids used this pheromone called E-beta-farnesene as an aphid alarm pheromone. So the experimenters just injected it here to simulate what would happen. So essentially, one aphid would release this alarm pheromone in response to danger, and then the remaining members of the colony would disperse, as you can see here. So this is work done by Will Hentley here, switching systems here to large raspberry aphid. And he did an experiment, it's slightly complicated, but maybe this diagram will help, where he essentially grew aphids and plants under ambient CO2 and elevated CO2, and then he did a sort of reciprocal cross where he moved aphids from ambient CO2 conditions to plants that have been grown under elevated CO2 conditions. And then with these different sets of plants and aphids, he did these sort of behavioral assays. So first of all, he presented these aphids with predator, a ladybird larva. With another group, he presented the aphids with the alarm pheromone. And in a third, he just used a control which had been used for the pheromone. And what he did is he recorded the escape response of aphids under ambient and elevated CO2 conditions. And this is essentially what he found. So in this first bar here, this is aphids that are being grown under ambient CO2. So you can see about, when they presented with an aphid predator, about 60% of those aphids will mount an escape response. But those aphids that are grown under elevated CO2, basically the escape response is less than half. It's about 25%. So those aphids are really showing impaired escape responses, essentially. And what's interesting is if you move aphids onto plants grown at ambient CO2, you see exactly the same pattern. So it's quite a persistent effect. And also it's a rapidly induced one, because if you move aphids from elevated CO2 to plants that have been grown under ambient CO2 plants, again, you see this impaired escape response. This is what happens when you do exactly the same thing with alarm pheromone. We see almost exactly the same pattern here. The numbers are lower, but typically that happens because you don't have the visual stimulus of a predator coming towards you. So Will and I sort of still working on ideas for why this pattern might happen. We speculate that it might be to do with compensatory feeding. If the aphids have to feed more intensively, it's actually difficult to stop that feeding to mount an escape. But we do know that similar patterns have been reported by other groups in 3 or 4 aphid species, so it seems to be a common phenomenon. Okay, moving on to the very final part of my talk, it's to do with aphid ant mutualisms. About 40% of aphids undertake this mutualistic relationship with ants. And because aphids feed on phloem sap, they have to take in a lot of phloem sap, which is low in nitrogen. And as a result, they accumulate a lot of sugar, which they then need to excrete, which they do as these kind of honeydew droplets. And a lot of ants have actually evolved to farm this honeydew. They've got a sweet tooth, essentially. And what they do is they work their way through an aphid colony and basically harvest these honeydew droplets, basically. It's a two-way street, though, because in return for providing ants with this honeydew, the ants will actually act as kind of bodyguards or sentinels, and they will attack natural enemies of the aphid colonies. So in this case, two ants are basically just attacking a ladybird until it, well, just to harass it until it goes, essentially. But it's like all mutualisms, it's kind of a sliding scale. So when the reward value, the honeydew value, is quite high, you can imagine that it's a very mutualistic relationship, so the ants are acting as bodyguards. But as soon as that reward value decreases, you can imagine that the ants actually see the aphids as a resource in their own right, so actually as a source of protein. So the bodyguard actually becomes the assassin in this relationship. And in terms of CO2 effects on plant quality, you can imagine that if nitrogen is low, then they've got to actually, the aphids have got to process more phloem sap and produce more sugar, so the reward value is going to be high. But if CO2 causes a decrease in nitrogen, then there's going to be less sap, less sugar, and you can see that reward value is likely to decrease. And this is what a master's student of mine discovered. What she found is that amino acid concentrations decreased under elevated CO2, but what happened was essentially was aphid population size didn't decrease, which is surprising given this decline in nutritional quality. But what she found is that under elevated CO2, the aphids were feeding a lot more intensively. So in theory, what should happen is that they're producing more honeydew, and indeed this is exactly what they did. So under elevated CO2, more honeydew was produced and droplet size increased. And as we would predict, ants actually became more mutualistic. They visited these aphid colonies much more and were incredibly aggressive towards their natural enemies. So just take-home messages, CO2 and temperature can act interactively, but they specifically affect functional groups of amino acids. It's not just amino acids altogether, at least in the lucerne system we were working on. Aphids can become insensitive to alarm pheromones under elevated CO2. This is kind of silent scream phenomena. But also atmospheric change can alter insect behavior. You know, the ants in our example became better bodyguards. So I'm just about out of time, so I'd just like to thank these people and thank you for your kind attention.
Brilliant, thank you, Scott. Thanks for keeping to time. Time for one quick question, I think.
Were those amino acids whole leaf or phloem sap?
Yeah, what James did is, so he did two different sort of extraction techniques. He used a sort of EDTA, so he looked at that, and then he compared it with amino acids that were basically in the tissue, and he found a really tight correlation. So in the end, we were able to just use the amino acids which were in the tissue, not necessarily the phloem sap, which is kind of incredibly difficult to obtain sometimes. So you got the correlation, essentially.
Great, thanks very much, Scott, and thanks for keeping to time again. Our next speaker then, who's Albert Passaradonna, and he's going to be speaking about the indirect effects of climate change on ecosystem properties as mediated by shifts in species dominance. And, oh, wow.
Seconds to go, yeah.
Until it turns over on the 15 minutes. Those of you who are regulars at the BES meeting will know that if people start sort of deviating by a minute or two, it all gets horribly messed up. But
All right.
try and jump between them. But over to you, Albert.
Yeah, thank you everyone so much for coming. So as this photo clearly shows, climate change has pervasive impacts on all aspects of life on Earth. This paper that came out recently did a really good job of summarizing most of these impacts, and they showed that they are very broad and they range from genes all the way up to ecosystems, biomes, and of course people. So when it comes to climate change impacts on ecosystems, the impacts can be direct. So for example, when persistent warming triggers the bleaching of coral reefs, or indirect as well. So when climate change changes the way a species interact with each other, or reorganizes community structure. And today, I would like to talk to you about these changes in community structure that arise from rain shifts. So one of the most commonly observed species responses to climate change is shifts in the distribution of species to track optimal habitat conditions. So as a result of this, for example, in the upcoming decades, we expect major changes in the vegetation of the world. So for example, in Spain, in northeastern Spain, where I'm from, we would expect temperate beach forests, such as this one, to be replaced by a warm Mediterranean vegetation. The thing is that actually most of these predictions are already a reality. So for example, this is a study that from Spain that shows that this is already happening, but there are many others. So as a marine ecologist, I'm much more interested in the forests that are underwater, and that is the forests that are formed by seaweeds. Seaweeds, just like trees on land, are also shifting their distributions in response to climate change. So in this recent review by Sandra Straub, we can see that some seaweeds in green are expanding their ranges in response to climate. Some other seaweeds are contracting their ranges in response to climate, and on top of that, all the blue dots are introductions. And as you can see in the UK, it's a pretty nice location because we have lots going on. So in your typical UK rock pool, you'll find some climate winners, such as this species, some climate losers, such as this one, and also invasive species. So it's a really good mix of a really interesting system to study. So what I study is the biggest seaweeds of all, so kelps, and I basically focus on two species. So we have this southern species that is found from Morocco all the way up to France, and now, because it extended its range in the UK as well. And then this other species that looks very similar, but is a cold water northern species. It's found from Portugal all the way up to Norway. So again, here in the UK, we have that since this species has extended its distribution, now it's replacing actually the northern one, to the point that in some locations it has become absolutely dominant, such as in this photo. So most of the work we try to do with my colleagues is understanding how these changes in seaweed composition will affect the functioning of the kelp forest. So some of the people I work with look at all the different assemblages that are associated with this kelp. So for example, they'll look at all the animals that live in this holdfast, these root-like structures that attach the kelps to the substrate. And they have shown that typically the southern species tends to support less biodiversity than the northern species. But myself, I'm much more interested in function. So to me, these kelp forests look a little bit like this. This is actually a car. So all these different pieces that work really well together and then provide humans a function. A car drives you places. So what I'm interested in is understanding what will happen if we replace the engine of the car by another one. Will the car still work? Will the car drive you places? So in other words, I'm interested in knowing if these novel forests function in a similar way than the ones before, and then understanding why, which is probably what you'll be most interested, because I'm suspecting there's not a lot of kelp lovers in this session. So what I'm focused in is the processes that are associated with carbon and nutrient cycling. So I look at things like productivity, trophic dynamics, decomposition. So kelps are extremely productive. Some of this productivity is consumed by grazers like these ones, and the rest is exported as detritus out of the system. And then this detritus is eventually also can be grazed or is eventually decomposed. And actually most of the productivity, around 80%, follows this route. So if we have a look at the productivity, this is the northern species. Here we have productivity, grams per day. Here we have the time. And this is the productivity at our 2 locations in the southwest of England, in Plymouth. So this northern species tends to grow early in the year, then peaks in the spring, and then ceases its growth and stops completely by summer. If we have a look at the southern species, it's a completely different story. This one, we have growth throughout much of the year, and then this one peaks in summer. The big difference here is that the northern species is what it's a textbook example of a season anticipator. So it starts really growing really early in the year in response to the day length. So its growth is not really well correlated with environmental factors, whereas the southern species, it's a season responder, so its growth is really correlated with environmental factors such as mean irradiance. So if we plot the mean monthly irradiance, we see that it tracks the growth curve of these southern species really well. And if we do a correlation, there's a really good correlation. So part of the differences in productivity between the 2 species is because they have this different growth strategy. Some of the factors that I think are also playing a role in determining their competitive ability are some key traits that differ between the species. So, for example, typically the southern species tends to have a higher SLA, so this probably gives allows it to capture more light. And again, also it tends to have a longer stage. So here we can see that it raises above all the other the northern species. So it's like forming a 2-tier canopy, and it's probably capturing more light. So let's have a look at what happens to this productivity, and we'll start by the grazers. It seems to be that the southern species tends to support or suffer from greater herbivory. So here we have the abundance of this herbivore per plant, this sea snail, and again we have 2 sites. In the yellow is the southern species, and the purple the northern species, and we can see that generally the southern species tends to support more grazers. If we have a look at another grazer, we see a similar thing. So once this limpet settles from the water column after May, again it seems to be that we find more grazers on the southern species. And then we try to have a look and try and understand why this may be, and we just did some simple feeding choice experiments. So basically we had these kelp biscuits, put a snail in the middle, leave it 2 days, and then measure how much of the biscuits have been consumed. So confirming our suspicions, it seems to be that the southern species is a preferred food. So the proliferation of these southern species seems to be that it will strengthen the trophic pathway within the forest, meaning that a greater part of this productivity will be channeled through this pathway, because the southern species is a preferred food and supports more grazers. But if you remember, I said that most of the productivity actually is lost as detritus, so we had to have a look at that as well. So here we have the erosion. So this is basically how much of these lamina tips get eroded per month, again grams per day. And in the northern species, again similar story. It suffers from greater erosion in spring, then remains relatively low, and then here we get another peak because of severe storm disturbance. So the southern species, completely different story again. This one suffers from more erosion towards summer, and that is because it's the time when it's reproductive, so it loses its spores, then the reproductive tissue becomes necrosed, and then there's this massive loss of biomass. And again, this peak relating to the storm disturbance. So then we tried to have a look at what happens to this detritus once it's exported, and we did this standard litter bag experiment. So basically pack some kelp into some bags, leave it for a month anchored to the seafloor, and then retrieve it, see how much it has lost. We did that at 2 sites, and at both sites it seems to be that the southern species decomposes faster than than the northern species. And again, we think this has to do, just like the feeding, with the biochemical properties and nutritional quality of the species. So even though they look quite similar, these two species seems to taste quite different. So I always say it's a bit like vegan bacon and real bacon. They look quite the same, but they taste differently. One taste is great, the other one not so much. So in conclusion, these novel forests seem to function quite differently. However, the core ecological processes like productivity were maintained. And again, these changes in function seem to be underpinned by key differences in plant functional traits. Now, of course, this is not new at all. So, for example, these rapid changes in novel plant communities have happened before. So, for example, in the Paleocene, Eocene thermal maximum, about 60 million years ago, there was also this short period of really rapid warming that featured also large and rapid range shifts in plant communities. And these plant communities were a mixture of native and this range shifting species. So just to make this point, here we would have the assemblage before and after, and then in this transitional period, we see that it's very different from anything else, and it changes quite rapidly. So this is not new. What is new is that now the humans are intrinsically linked to all these ecosystems. And so every potential impact on climate change impact in these ecosystems has the potential to impact ecosystem services and impact humans. So I would just like to finish up posing some questions I need to think about. I think we need to think about when we work with these novel plant communities. So how do these novel plant communities affect ecosystem services? We have this these species coming in, coming out, but I think we need to be more focused and really looking at what impacts will result for ecosystem services. Then, of course, how can we manage these novel communities? Even though they may be transitory, this is like at least 2,000 years, so we need to manage these communities, and then if we can predict the impact of ecosystem reorganizations by looking at the change of these key functional traits. And with that, I would just like to thank you, and thank you to all these people as well. Thank you.
Great, thanks very much. We're bang on time, so we've got time for one, maybe two questions. There are any?
No.
What's happening above the food chain following these grazers? So you've got this big explosion of grazers.
Right.
Are we seeing
Yeah, the thing is,
something else happening above that?
yeah, this is something I meant to look at. Some of these grazers, I've done some experiments and do get consumed by fish. The only thing, it's a bit tricky now, is I don't know if these grazers are already inside the forest, and then, since this species is more palatable, they come up, and then this makes them more conspicuous to predators, and then maybe it would translate, there would be a predator effect. Or I don't know if these are already in the forest and just like the fish don't see them.
There's one quick question. We've got time. Otherwise, I think we'll probably move on. But thank you
All
very
right.
much.
Thank
Great
you.
talk and good timekeeping. So our next speaker then is Danny Rabaiotti, who's going to be talking about the evidence of increased mortality at high temperatures in a tropical mammal, the African wild dog. And we're going to wait until... Yeah. just to follow that up, I mean, in a lot of Africa, the actual basic climate data are few and far between. I mean, do you think we know well enough what the baseline temperatures are at the moment across the range of the wild dog?
So yes, this is definitely a challenge. The current data sets, climate data sets that are out there are not great. But I think we've got quite a few long-term study sites on the wild dog. That's why it's such a great species to work on. With quite a few of them have weather stations as well. So I've been able to check at least like my study site against current climate data. And a lot of it isn't great, but some of it is okay. And I think, okay, there are flaws, and there will always be flaws in climate data, especially for Africa where there's such low coverage. But you've just got to work with kind of the best you've got, I guess.
Yeah, and it can be improved, obviously.
Yes, definitely. And I think it's really important that we get more weather stations out in Africa so that those climate layers can be improved and that we better link up any weather stations with these geographic climate layers.
Sure. Was there another question up the top I saw? Yes. Oh, it's gentleman in the back row, and then you.
Those two might ameliorate each other. So there's some of these studies coming out showing that in some cases those two can interact, and a larger group can buck you against the effect on the temperature, or vice versa.
Yeah, so I think one interesting thing to note about group size is when they disperse, they're in much lower group sizes, and actually the group sizing actually overrate dispersal, so it is the low group sizes that are the really important factor. But no matter, there was no interaction between the two, so I would say probably not from the evidence that I've got.
Okay. Sorry, down there. Yeah.
So you showed about the high temperatures affecting individual deaths. Just what impact that had on packs, and whether there were cases where you lost whole panels due to temperature.
So we can't say, oh, it was definitely temperature that caused this, but there has been incidents where we've lost whole packs, for example, to disease, to human causes, and those sorts of things. And I think it's not, I'm not saying, oh, temperature caused them to die. It's just leading to them to be more likely to die of these other factors. It's not like it's hot and then the wild dog just drops dead. It's more a case of they're probably coming in more into contact with dangerous situations, and it's probably any diseases they already have making it worse. So yeah, I wouldn't say temperature definitely caused any wild dog deaths, but what I would say is just from these results, it's probably exacerbating other factors that cause wild dog deaths.
Okay. Pete, yeah, I think.
I mean, you've got all the data to show whether or not in that preceding month they hunt less, presumably because you've got. Is that the case?
For some of the dogs it is. I already did a paper basically on showing that activity levels in wild dogs, whilst they're lower in the day, they do try and compensate at night for that. So they're more active at night when it's hotter in the day. However, they can't compensate for that when they're denning. They don't become more active. So there seems to be this limiting factor on that. But because we've just got activity centres, and those are only on some of the GPS collars, I can't link that with every dog, because, for example, the VHF collar dogs don't have those activity centres on them. And also in our study site, because they eat such small prey, we can't be like, oh, there's a wild dog eating, because it's just kind of, it's a little bit more difficult to track. We can't track them down and kill because they eat tiny amounts of
Okay, we do still have time for another question, if there is one. Oh, yes.
Thanks for a great talk. When people think about climate change impacts on wildlife, often they think about they think about polar bears, high melting ice. There's a wealth of, obviously, most species are in the tropics. Do you have any clue of why it is that wild dogs are particularly vulnerable, or are there other tropical species that we should be worried about?
I think because of their activity patterns and the fact that they are
Okay, brilliant. Thank you very much. And we'll move on now to, yes. So our next speaker then is Elizabeth Morrison, and she's going to be talking about species traits alter climate change threat assessments for European mammals.
Okay, hi everyone. So there's a vast amount of research which use models to predict the impact that climate change is having on species. But emerging research is now using species characteristics to help identify which species will survive climate change. So my research with Exeter University uses traits which relate to species' ability to reach climatically suitable areas in the future to more accurately identify which species will become threatened. So as we all know, climate change is forcing species to move to new areas of suitable habitat when climate conditions become intolerable in their current range. So this includes movements in elevation and latitude, which can cause increased extinction rates when future habitat is too small or too fragmented. So therefore, it's vital that we identify future climatically suitable areas and understand species' ability to reach these areas to help prioritize conservation efforts. So the most frequent method for forecasting species exposure to climate change are species distribution models, or SDMs. So they correlate species' current distribution with various climatic variables, for example, annual temperature and annual rainfall. And then this is used to estimate which geographic areas will become suitable in the future, projected under various climate scenarios. So in this study, we used, to estimate the exposure that European mammals face under climate change, we used species distribution models to obtain 3 exposure indicators. So this is an example of one of the maps that I created for each European mammal in the study. And the grey areas on the map are those areas that are always unsuitable for the species, and the yellow areas are those always suitable for the species. So the first exposure indicator is the area gained by a species, which is the green areas. So this is the area that is climatically unsuitable at present, but predicted to become suitable in the future. So areas that a species can move to under climate change. The second indicator is the area lost, so the red areas on the map. So this is the area that is climatically suitable at present, but predicted to become unsuitable in the future. So areas that a species could lose under climate change. The final indicator is the range margin shift, shown by the black arrows. So this is the distance that a species is required to shift in order to reach climatically suitable areas in the future. So the distance a species needs to move in order to reach the green areas on the map. So that makes up the 3 exposure indicators. But there's a key problem with species distribution models. They don't take into account whether species have the ability to reach these areas in the future, which is also termed a species range shift capacity. So species might be predicted to gain a large amount of habitat in the future and therefore be less exposed to climate change. But if they don't have the ability to reach these areas, then they're still threatened by climate change. So in order to account for species range shift capacity, emerging research is now using species traits. So examples of traits that are used in the literature are dispersal ability and diet breadth. So if a species has a poor dispersal ability and a very specialized diet, then they'll be less likely to colonize future areas than a more generalist species. So such traits can be used to provide an estimate of the vulnerability that species face. But many traits that are selected for studies don't actually correspond to species range shift capacity, and this causes many important characteristics to be ignored. So Estrada et al. Came up with a predictive trait framework, which splits the range shift process into 4 key stages. So you have emigration, movement, establishment, and proliferation. And then using existing evidence, identified 7 trait categories which correspond to each process. And this allows the traits to correspond to species range shift capacity. So traits relating to site fidelity correspond to species' ability to emigrate. Then traits relating to movement ability to species' ability to move to new areas. And then avoidance of small population effects and persistence under unfavorable conditions correspond to species' ability to establish in new areas. And then competitive ability, ecological generalization, and reproductive strategy correspond to species' ability to establish and proliferate in new areas. So in the current study, we selected 2 of these range shift processes, establishment and proliferation, and 2 of the trait categories, ecological generalization and reproductive strategy. And these were selected because it's been shown that they are the most important determinant for a biogeographic proxy of range shifts. So the 2 trait categories ecological generalization and reproductive strategy, were used as the two vulnerability indicators in the study, and these correspond to species' ability to establish and proliferate. So each of these indicators can be informed by 3 traits each. So ecological generalization can be informed by species brain size, diet breadth, and habitat breadth, and then reproductive strategy can be informed by species litter size, interbirth interval, and sexual maturity age. And the 3 traits in each category were combined to give 1 value for ecological generalization and 1 value for reproductive strategy. So the overall aim of the study was to combine commonly measured exposure indicators from species distribution models with more rarely measured vulnerability indicators from traits to provide a risk assessment of the threat that 37 European mammals face under climate change. So to do this, we created risk syndromes. So this is an example of the risk syndrome that we used for each species. These are the 3 exposure indicators on the top and the 2 vulnerability indicators on the bottom. So EG is ecological generalization and RS is reproductive strategy. And then each of these indicators was assigned what we called a threat level, which ranges from 1 to 10, as you can see on here. So if a species had a low threat level of below 5, shown in the blue area, then a species had low exposure and low vulnerability. So a species with low exposure would have a low amount of area loss, a low range margin shift, but a large amount of area gained. And then a species with low vulnerability would have high ecological generalization, so be a more generalist species, and have a high reproductive strategy, so have a high reproductive output. And then species with a threat level of 5 or above, shown in the red areas, would have high exposure and high vulnerability. So high exposure would be a high amount of area loss, a high range margin shift, but a low amount of area gained. And then high vulnerability would be low ecological generalization, so more specialist species, and low reproductive strategy, so have low reproductive output. And of course, a species can have high exposure and low vulnerability, and low exposure and high vulnerability. So each species in the study was assigned to one of these 4 categories, shown on the x-axis. So on one side, we have high exposure and high vulnerability, and on the other end, low exposure and low vulnerability, with combinations of the 2 in the middle. And then we have this percentage of species on the y-axis. So we found that nearly half of our study species, 41%, had high exposure and high vulnerability. These are the species that are the most threatened under climate change. So they're threatened by their exposure, so they're going to lose a lot of habitat, gain very little, or have to move very far. And they're threatened by vulnerability, so they possess traits that prevent them from establishing and proliferating in new areas. And then 24% of species still had high exposure to climate change, but had low vulnerability. So these species had traits that enabled them to establish and proliferate in any available areas. And then 16% of species had low exposure and high vulnerability. So they should be able to gain enough habitat or not have to move very far. But they possess traits that prevent them from establishing and proliferating in these available areas. And then only 19% of species had low exposure and low vulnerability, so the species that are the least threatened by climate change. So now I'll show you a few key examples of species to show how our risk syndromes could alter traditional risk assessments like the IUCN Red List. So the first species is the Eurasian elk, and this is its risk syndrome on the right. And as you can see, the species has a high threat level for all of the indicators apart from range margin shift, which is about 1. So this species is predicted to lose a lot of habitat, gain very little, and has low ecological generalization and low reproductive strategy. So this species was classed as being high risk, so having high exposure and high vulnerability. Then the IUCN European Red List has classed the species as least concern. So although the Eurasian elk may not be threatened at present, we're predicting that under climate change, this species is going to become highly exposed and highly vulnerable. The second example is the marble polecat, and apart from having a high threat level for range margin shift, so having to move quite a large distance to reach climatically suitable areas, it's relatively unthreatened by climate change. And we've classed this species as being low risk, so low exposure and low vulnerability. The IUCN Red List classes species as being vulnerable to extinction. So although we've predicted that this species isn't threatened by climate change, there are most likely other factors like habitat loss that are causing this species to decline. The last example is of the wolverine, and as you can see, the species has a threat level of 5 or above for all 5 indicators. So we've classed this species as being high risk, so high exposure and high vulnerability. The IUCN Red List also classes species as being vulnerable to extinction. So the wolverine is already threatened by fragmented distribution in Europe, and we're predicting that it's going to become even more threatened, so have high exposure and high vulnerability under climate change. So the main take-home message of the study was that the majority of species were threatened by both exposure to changing climates and by vulnerability, so possess traits that prevent establishment and proliferation in new areas. We've also shown that the inclusion of traits that relate to range shift capacity could alter previous threat assessments and could cause threat classifications to starkly change under climate change. But we must not discount the validity of species distribution models, and combining both exposure and vulnerability metrics is of great value for creating risk assessments. So future studies should focus on research more into traits, so focus on traits relating to species' ability to establish and proliferate like we have, and also species' ability to move if there's sufficient data to do so. So recently, the IUCN have developed best practice guidelines for assessing species vulnerability under climate change. This includes methods on using species traits to produce vulnerability assessments. So the case studies in the guidelines vary from African amphibians to reef-building corals and butterflies and everything in between. So this is an excellent step that will hopefully allow more studies like this one to more accurately identify at-risk species, to develop effective conservation plans and help reduce global biodiversity loss. Thank you for listening.
Thank you, Elizabeth. And another example of perfect timing, so we do have time for some questions. I'm going to ask one first, a bit cheeky as chair. One of the other big limitations of species distribution models is the extent to which present distribution reflects climate as opposed to history of management or fragmentation and other things. Did you do any assessment of whether your 33 species were present distributions were climate limited?
No, we didn't. I was actually given the data already, so I put it into GIS, so no, I haven't done anything like that.
Yeah, okay, that's it. Maybe the next step.
Yeah, yeah.
Okay, up there.
Yeah, you classify some species as not being very vulnerable because they have a great range expansion, even though they're very vulnerable according to the traits. But the fact that they have a high area or a high amount of area they can expand or move into doesn't mean that they actually can with habitat stratification. Did you take that into account at all?
No, we didn't. No, we just looked at, it was more about using this new framework, so making sure that the traits that are used actually correspond to range shift capacity rather than traits that, you know, don't show that necessarily.
Oh, right, lots of questions. If we can make them quick, we'll see how far we get. You, I think, you're first. Put your hand up.
Yeah, so the marble polecat was for habitat loss, and then the wolverine was because of the fragmented distribution. So none were actually talking about climate change.
Okay, next line down.
Yeah, that was because there just wasn't enough data for European mammals. So for direct traits, so like their dispersal distance, there was about 4 or 5 species that we actually had data for, so there was just no point including it.
Okay, Danny, if it's quick.
Really quick, I was just wondering if there was any species you looked at within those species that other papers had said, oh, this might be threatened by climate change. You know, were you surprised by any species?
No, there's not that much done on mammals, not with traits anyway, so it was quite a new thing. Most is done with birds and plants and amphibians and things, yeah.
Brilliant, thank you very much. Okay, now for something completely different, I think. The impacts of mid-Holocene drought on Bolivian seasonally dry tropical forests. And Heather Plumpton is our next speaker, who will start when that...
When the buzzer goes.
goes to 11 o'clock precisely. There we go. Over to you.
Hi. So I'm Heather, and I'm going to be talking to you about some of my PhD work on the impact of mid-Holocene drought on Bolivian seasonally dry tropical forests. So firstly, I thought I'd tell you a little bit about this ecosystem. So they're quite different to the evergreen, humid rainforests of Amazonia. And floristically, they're quite different, and they also tend to be semi-deciduous and shorter in stature. This map here shows the distribution across South America, and the distribution is controlled primarily by climate, and they occur, not surprisingly, in places where the climate is dry. So typically they have a long dry season, 5 to 6 months of the year, with precipitation lower than 100 millimeters. But the distribution is also controlled by edaphics, so they tend to occur on fertile soils. So where climate is dry, but the soils are poor, you often tend to get dry savannas, which produces mosaics of dry forest and dry savanna within a landscape. So why are we interested in studying tropical dry forests? Well, firstly, they're hugely understudied. They're often passed over to study the more charismatic evergreen forests of Amazonia. And they're also hugely threatened. So I mentioned earlier that they grow on areas with fertile soils. So these fertile soils are very suitable for agriculture. So tropical dry forests tend to be highly deforested and fragmented ecosystems. And in fact, only 10% of their original cover is remaining in the neotropics. And they're one of the most threatened tropical terrestrial ecosystems. And due to the fact that they're understudied, we're not sure what the impacts of future climate change are going to be on these forests. So some models are predicting increased drought for the region with future climate change, and these forests are potentially vulnerable to those changes, but we just don't know the extent or the impact. So why study a drier climate that happened 6,000 years ago? So we know one way of looking at the impact of when we're working out what the impacts of future drought are going to be, is to look at what the impacts of droughts in the past were. So we know that there was a drought around 6,000 years ago in tropical South America, which is the mid-Holocene period, due to lake low-level stands at Lake Titicaca, this record, and dust concentration records, and further pollen records, reconstructions from across South America. So we can use this time period as an analog for what may happen to forests under future climate change. And this allows us to study the real long-term effects that climate changes can have on forest dynamics on the scale of hundreds to thousands of years, which can't be achieved by shorter-term ecological studies alone, and maybe allow us to test the predictions that models are making on the scales of hundreds of years. So my research question is, what are the long-term impacts of drought on tropical dry forests? So some basic hypotheses or potential outcomes of my work are the possible best-case scenario: the forest is resilient to the drought, there are no floristic changes, it's wonderful for conservation, everything survives. To the worst-case scenario, where the forest is very sensitive to drought conditions, the canopy opens up, and we see an encroachment of savanna species and a reduction in forest area. And then there's the middle ground option, where forest is resilient as a biome, but there are some floristic changes within that. And then my second question relates to the ecotones between dry forest and dry savanna that I mentioned earlier, and what really controls this ecosystem, this ecotone, and how it responds to drought. So the ecotone is thought to be controlled by edaphics and the quality of the soil. What I'm really interested in working out is whether climate can override those edaphic constraints. And if it does, then during this dry period, we might expect to see a savanna expansion at the expense of dry forests. So the savannas are able to move from the poor soil areas into the more fertile areas as the forest dies back under drought. But if climate cannot override edaphics, then the ecotone will be predicted to be stable with no movement. So the approach that I'm taking is a paleo approach. I'm a paleoecologist. My lake site is Laguna Mandioré in Bolivia, South America. And we've taken a 4.5 meter deep lake sediment core from this lake, which I'm using to reconstruct vegetation changes over the past 10,000 years using multiple techniques. So microfossils, fossilized pollen, and phytoliths, which are silica bodies produced by plants, which are preserved in sediments, and also some geochemical proxies, carbon-nitrogen content, and stable carbon-isotope ratios of the sediment. So where is my study site? Well, this is obviously South America, and the black box here highlights the region that I'm working in. I'm on the border between Bolivia and Brazil, so the Rio Paraguay is effectively the border here. So my lake site is surrounded primarily to the west by seasonally dry tropical forest, marked on this map as SDTF, to the north and the west, and a little bit to the south. And then to the northeast, we have this hill formation called the Amola Hills, where soils become poorer as you go up the hill, and dry forest grades into upland savanna. And then to the west, we have the Pantanal wetlands of Brazil. So a couple of key dry forest taxa to make you aware of that I'm going to be mentioning later. Astronium is a key dry forest tree, as well as Anadenanthera. And up in the upland savanna, we have Curatella americana, which is a tree that you only find in open savanna environments. And then herbs such as Boreria, which are typical of open savanna environments. So, what does my record from Laguna Mandioré show? This is my fossilized pollen record. For those who aren't paleoecologists and not familiar with pollen records, I'll just give a brief explanation. So on the y-axis here, we have depth in centimeters going down my core. And next to it, I've got 2 dates, which are radiocarbon dates in calibrated years before present. We're applying for further dates from NERC. So eventually this axis will represent time. The deeper you go, the further back in time you go. And then along the x-axis, we've got various pollen taxa, which I've identified in my record, and how their abundance varies through time. So these axes here are percentage abundances. A couple of key taxa are highlighted in bold. So Astronium, as I just mentioned, is a dry forest taxa. Anadenanthera, another key dry forest taxa. Curatella there is the savanna tree. And then I've separated out the herbs here, so Boreria and Alternanthera are key indicators of open environments. So what are we seeing? Well, around 6,000 years ago, the mid-Holocene drought period is roughly 200 centimeters here. And we can see that the dry forest indicators are present, and they're actually increasing during this period. Here, Astronium and Anadenanthera. Alongside some other general forest taxa, such as Moraceae, Melastomataceae, which are consistently present throughout the record. But we also see an increase in Curatella, the savanna tree, and some increases in herbs for open environments. So potentially we're seeing the dry forest is being maintained throughout the drought, and in fact, key drought-resistant taxa are increasing, but we're also seeing an expansion of savanna. So this basic schematic explains what this vegetation change might have actually looked like. So we start pre-drought with these green trees, which are sort of generalist tack forest. The brown tree is dry forest taxa and herbs. So pre-drought, we've got generalist forest taxa dominating the forest assemblage with some herbs on top of the hills. And then during the drought period, we see forest dominance overtaken by key dry forest taxa, drought-resistant taxa, concurrent with an increase in savanna indicators, an expansion of the savanna, possibly downhill into previously forested areas. And then post-drought, we've got a decline in savanna indicators, so savannas are now restricted again to the top of the hills, and a co-occurrence of generalist forest taxa and dry forest taxa. So in terms of my original questions, the impact of drought on dry forests seems to be some level of biome resilience, but with floristic changes in there as well. So key increases in drought-resistant taxa. In terms of question 2, the dry forest savanna ecotone under drought seems to be an increase in savanna at the expense of forest, with potential climatic controls overriding edaphic constraints. But these changes are only local in scale, and they only occur at the ecotones, as evidenced by the resilience of the dry forest biome in the area. So potentially good news for conservation of this biome, some biome resilience. As I've got a lot more work to do on this site for my PhD, but for now, I'd just like to thank my supervisors and collaborators, and thank you for listening.
Great, thank you, Heather. So, time for some questions.
Any of the, sorry? I haven't done phytoliths yet, so yeah, that's the next thing on my list, along with charcoal, which I didn't mention as well. But yeah, that's going to be key to separating out these savanna changes.
Okay, a couple of questions. Yes, there.
Sorry, I couldn't quite.
Sorry, what's that? Are you planning to macro micro charcoal to look at anything from drier or human disturbance in the regime shifts?
Yes, so that's another proxy that should have been on that slide, that I am planning to do charcoal to reconstruct the fire regime. Yeah, particularly, I want to look at the role of how drought changes the role of fire within the dry forest. But also, the role of human disturbance is going to be interesting. So there have been no studies that have found humans in this area during the time period that I'm looking at, but that's obviously not to say that they weren't there. So I'm going to be looking at the charcoal and also the phytoliths, because you can get crop taxa identified in the phytolith record, which will help me identify whether humans were there and whether they were helping to drive these shifts. So yeah.
Okay, I think there's a question 2 rows up.
Oh yeah, it was just a quick one about Astronian.
Yeah.
So did you identify it as a species or only at genus level, the pollen of Astronian?
Yeah.
Because it's a rather broad genus, and to use that as an indicator for dry forest, because there's some crossover in Astronian between dry and wet and semi-wet forest.
Yeah, that's a good point, and this is always a problem with pollen, using pollen indicators. Yeah, we can only identify it to genus level, but the argument that we would use is that because we know that that species of Astronium, which is found in dry forest, is around that lake site, it's most likely to be coming from that species of Astronium, but we can't rule out the possibility that some of it's been blown in from evergreen forests that are they're hundreds of kilometres north, so it would be a small...
Astronium arundeula, I think, is it?
I can't remember the name, but yeah. So.
Thank you.
I had a question, which is around the topography and the local variations in the land and the forest there. I mean, is there the possibility that, perhaps on lower lying, more damp places, that the seasonal forest could locally persist, even if, more broadly the savanna starts to encroach?
Yeah, I mean, the topography around here is very interesting. So the little schematic map that I showed probably didn't give as much information as it could, but to the west of the lake, there's the wetlands of the Pantanal, which includes lots of islands, and then depressions in which there's wet savanna, and then islands, which dry forest tends to persist in even when it's dry.
Yeah.
So termite mounds and other formations where they can persist locally,
Any other questions? No? Okay, well, thanks very much. It's a really nice study system, I think. Thank you. Okay, moving on then. Our next speaker is Andre is going to be talking about short-term stress memory of Plantago lanceolata after drought and frost exposure. As before, we'll just wait until the clock turns over to 15. It's one of the nice things about this session is the range Montego, I'll see you later. Over to you.
Thanks. Yeah, so first of all, yeah, I'm going to take you from, we've been now in South America, so I'll take you from there back inside the lab, and I'll basically compress our study period from 6,000 years to just about 3 weeks and try to convince you that that's also kind of interesting. So basically, we've just started a series of new experiments in our lab in Greifswald, Germany. It's close to Berlin. And these experiments all have to do with this phenomena of stress memory. So what is stress memory? Why is it interesting? Why do we care? Well, in the last 5 years, basically, there have been some studies coming out that have shown that grasses remain acclimated to drought, for example. So basically, if a plant experiences drought earlier on in the season or earlier on in the summer, and then encounters a drought again later on, then it may be able to better withstand the second one or not be as damaged as plants that hadn't had the first drought. So then, since then, around 3 years ago, these my co-authors, one of them being my supervisor at that time, Jürgen Kreiling, they've published actually a review on this. So basically looking at stress memory, and basically there they hinted that higher stability of ecosystems than previously expected may be due to ecological stress memory. So basically, if you have, let's say, a tree or a long-lived organism, plant species, if it experiences any kind of stress, let's say, sorry, drought or frost stress, then it may be able to actually cope with it way better the second or the third time around. So it's important to study this phenomena and just explore just how great of an impact it can have. And then very recently, this just a paper came out in this year from Canada that basically have shown that in this case, this grass, Poa pratensis, when it experienced frost in the winter, it was able to cope with summer drought better than the plants that hadn't been exposed to this frost stress. So our hypotheses were basically that because frost and drought stress are not literally, but basically physiologically similar in that water is drawn out from the cells, so it's kind of a dehydration stress, you can think of it that way. So we hypothesized that previous exposure to drought would enhance subsequent drought and frost tolerance, and also previous exposure to frost would basically do the same. So that was just our hypothesis. And how did we do it? So basically, we had Plantago lanceolata was our study organism, and we had 7 different scenarios for our plants. So first, we reared all of them under similar conditions until they reached these approximate parameters seen above there, and then the control had nothing. Then we had either plants that had just the drought or frost when they were 80 days old, and then we had basically plants that had a first stress, being drought or frost, when they were around 60 days old, and then the second stress being either frost or drought, again when they were 80 days old. And so here it has to be mentioned that our drought was actually a combination of drought and heat stress, because we didn't want to wait for these plants are pretty drought tolerant, and it takes about a month if you even stop watering the pots to even see anything. So this was a master's project, and yeah, there was we needed to get results quick. So anyway, we warmed them up, and just in 4 days, we got basically the same results as in a month at 36 degree day temperature and 22 degree temperature at night. And the frost was basically a -5 degree frost for 4 hours. So what did we see? Well, the first, I guess, interesting point is that a point can be made here for cross-stress tolerance instead of recurrent stress tolerance. So what I mean by that, so if plants that had the same stresses or the same stress imposed on them twice, drought, drought, or frost, frost, so they fared the worst. And plants that had a combination, so either drought, frost, or frost, drought, so they were better. Obviously, control plants with no stress had the greatest biomass in the end. And here is also interesting to see that the plants that had experienced drought before frost, they were the only ones that were not significantly different from the control treatment in terms of their final biomass. So possible indication of a memory effect there. So we see here a similar pattern for spot measurements, just a measure of greenness of the plants at the end with less significance values that we see for our biomass values, but a similar pattern. And if we look at a couple of other parameters that we've measured, namely the change in leaf number and percent change in leaf length, again we see similar patterns as in the previous slide. Here, again, we see that especially for the change in leaf number, we see the biggest sort of hint that previous exposure to drought made the plants withstand frost better the second time around. So then we were just wondering, well, we wanted to, we thought, well, there's obviously lots of literature that you can have stress memory even after the first drought, even if the stress is the same as before. So we basically did a different scenario, and this time we basically increased the size of the stress when the plants were 40 days old. So instead of a 4-day stress period, we gave them a 10-day drought stress, and then we gave them a longer recovery period. So this time, basically, as you see here, from 40 days to 78 days. 78 days old is actually when we collected them, but they got a 4-day recovery period. Sorry, a 4-week recovery period instead of the 3-week recovery period as in the previous experiment. So we basically just played around with the magnitude of stress, the recovery period, to see if we could get on a very small time temporal resolution, if we could get this drought stress memory effect. So what did we see here? Well, first of all, it's interesting just to see that the smaller plants, even after we expose them to 11 days of drought, they basically reach similar levels of soil moisture level in their pots as the older plants when the second drought came around. So just because smaller plants, they transpire less than bigger plants, so they take a longer time to reach the same stress level. This time we also measured for every single plant, we measured their stomatal conductance. So here again, first we see that basically after our 11 days of drought, they reached similar stomatal conductance values as the plants, as the older plants after only 6 days. And now if we, here basically on the left-hand side, we have the stomatal conductance measurements of plants during the first drought. Here we have the same plants now having recovered after the first drought and now experiencing their second drought of 6 days. So here we see that perhaps potentially one could hypothesize or think that this may be sort of that they've learned to not keep their stomata open, and they already had less stomata open at the beginning of the second drought, giving us this relationship between the stomatal conductance and the days of drought. We can also we also measured soil moisture, so we can also plot the same changes in stomatal conductance versus the soil moisture, and basically we found that plants that hadn't experienced this 10-day drought in the beginning, out of the over 80% of the plants died. However, only 20% of the plants died that did experience the first 10-day drought. So we're not sure what to make of these changes in basically the slope being not as steep in the plants that had the drought before compared to plants that had never experienced a drought as the soil moisture decreased. We know that in the end, their soil moisture content at the end of the second drought was very similar, and still we had a higher survival rate of these plants. So this may be a key point that they started with, they lost overall less water perhaps by starting at a lower stomatal conductance at the start of the second drought. So in conclusion, so we had the two hypotheses. For the first one, was previous exposure to drought beneficial? Well, yeah, potentially beneficial after a 3 to 4 week recovery, and the cross stress was potentially more beneficial than recurrent stress. And for the previous exposure to frost, here we did not see memory effects after our very, very short, brief recovery periods. And what are the implications of this? Well, basically, of course, this was a very, you can't really extrapolate this much to the field just because of the sort of 3-week proximity periods between such a heat and drought stress followed by frost stress is not realistic. So this was more of a theoretical stress memory experiment. But basically, we've shown that in as little as 3 weeks, you can have this stress memory effect. And we've also shown that the order of these effects, their magnitude, and the recovery time all play a role in basically in how well they can enhance a plant's ability to withstand future stresses within its lifetime, depending on when and how big of a stress it had experienced. Yeah, that's basically it. So thanks a lot for your attention.
Thanks, Andre. Another good punctual talk. Questions?
Do you think the memory effects would vary across the species range? So areas where the populations are experiencing more fluctuations within a given year would show more propensity towards memory effects?
That would be a really interesting question. To be honest, I have not encountered this in literature, so I don't know. However, it's definitely the next direction to take. So also to take this to plasticity and to genotypes and phenotypes, and that's basically where we're going with this. So among species versus within species differences in the potential to show this stress memory effect.
Any other questions? I've got one. Did you look at rooting depth at all? And was there any response going on below ground?
No, unfortunately we didn't do that. But yeah, that's.
You could see one adaptation would be to, allocate more below ground.
Yeah, bigger, longer roots. Definitely.
Any other questions? Yes.
Did you ever consider looking into the molecular aspects of it, like doing temperature to the stress response to see the change of the
I think that's definitely where you have to go. We're a plant experimental ecology lab, so we like to work with as little in the lab and just with plants as possible. So I think where we will move with this is actually on making this more realistic. So sort of doing this outside, maybe with heating lamps, and then having natural frost, so to simulate this in field conditions. And I think maybe with cooperations, then we could do it with labs that do molecular and genetic work. Yeah.
Okay, thank you very much. So So our next speaker then is Martin Kay, another shift in location and habitat type, this time looking at ecosystem responses of coastal peatlands to seawater flow.
Thank you. Yes, so this is some of the work which is going to be contributing towards my PhD thesis, looking at climate change and lowland raised bogs. But for this work, I'm interested in coastal bogs. Bogs are ombrotrophic wetlands. They have generally higher precipitation than evaporation, very high water table, and a specialised plant community to exist in these conditions. It's recently been estimated as about 11,500 kilometres square of coastal wetlands below 2 metres above sea level, which are at risk of flooding from seawater. So latest IPCC reports are suggesting that the rate of sea level rise is increasing, but also storminess, so further problems with storm surges and tidal surges in these types of habitats. And we've lost a lot of bog, especially in the UK, through conversion to agriculture. It makes really good grade 1 agricultural soil, and also through development of horticulture, and also because it's a great place for vectors of diseases like malaria and for the mosquitoes to breed in. So a quick look at the bog ecosystem. So in the centre there, you've got the green and the brown there. Those are two species of bryophyte of the genus Sphagnum. And these are really important in these bogs. These are ecosystem engineers. So they efficiently intercept nutrients which are fed into these bogs from the atmosphere rather than from groundwater. They also affect the acidity of the water. They increase the acidity, reduce the pH, and they're also composed of rather recalcitrant materials, so they're difficult to break down. And because of those high water table levels, the anoxia, it's already rather difficult for microbes to get along as it is. So the carbon is taken out of the atmosphere by the plants. Part of that is used in metabolic processes and respired back to the atmosphere. Another part is taken up in plant biomass, and then some of that is decomposed, returned to the atmosphere. But because of the anoxia, the acidity, and that recalcitrance of the material, some of this carbon is then sequestered and it becomes the peat soil. However, there's also vascular plants in these habitats, and they have particular physiological adaptations which allow them to have an oxic area, an aerobic area around the rooting zone, and this also allows the transmission of methane, another important greenhouse gas, through to the atmosphere. So to look at how these gas fluxes will change in the event of seawater flooding, I've been conducting an experiment at Borth Bog, Cors Fochno, which is just north of Aberystwyth on the West Wales coast, adjacent to the Irish Sea in Cardigan Bay. So this is the general area. We've got a major river channel to the north there, and we've got the bog in the centre. This is an active raised bog, so it is sequestering carbon over time, and it's surrounded, the active area is surrounded by a degraded bog area. It's about 6.5 kilometres square. And the main threats from sea level rise and storm surges in this area come directly from the sea over a spit of land where a town has been built, the town of Borth. That town's about 5 metres above sea level. The bog behind is about 3 or 4 at the maximum height. There's also the big river channel to the north where seawater comes in at high tide, and then particularly at spring tides, it can rush in. There's a salt marsh area along here, and here we've got converted salt marsh and bog areas which are also quite low. So there's a threat of seawater rushing down over that area, and there's also a canal which has been cut through the bog for drainage back in the 19th century. That's higher than the bog, and it's also made of peat banks, so there's a good chance that could fail at some time. So in this experiment, I've got 15 plots, and they're divided into 3 treatments: seawater flooding, bog water flooding, or just control. Each plot is 33 centimetres in diameter. The tube's of 1 metre length, but there's only 90 centimetres within the peat column, and there's 10 centimetres standing proud of the surface there. There's also a tube for water table depth measurements, and taking samples for water analysis. So a one-off treatment was applied in April 2015, and that was 10 litres of either seawater or bog water, both collected locally. Plots like this full to the surface, and that lasted about a week before the level of the water in the plots had drained down to below where the sphagnum surface was. I've been taking measurements through to August this year, and I've been looking at the physical characteristics of the bog, so water table depth, the internal sphagnum surface height, temperatures, chemical conditions, pH, conductivity, and I'll also have some chemical data as well. The condition of the plant, so some plant physiology, and also percentage cover of species within the plots. And finally, and what really I'm going to talk about today, the gaseous side of things, so CO2, CH4 fluxes to and from the bogs, and I've been using a Los Gatos greenhouse gas analyzer for these measurements. So we'll go through some pictures first, just to see what the plots are like. I've only got to show the control and the seawater, because the bog water plots were very similar in appearance throughout the experiment. So this is April 2015, prior to treatments. I selected the plots in March, to have them rather homogeneous, really, in terms of the plant community. So the most of the green there is Sphagnum cuspidatum. There are some sedges within there, Eriophrym, and there's a bit of Sphagnum pulchrum in that one. And there's also birds which like to come along and throw sphagnum in plots as well. So I think some of that was due to them. In 2015, 3 months after the treatments have been applied, the control and bog water plots were looking perfectly fine and very similar to the external aspects of the plot as well. However, with the seawater, sphagnum had changed to a very dark green olive colour. When there was water in the plots, there would be small fragments of sphagnum floating around in there as well. And the Eriophrym, which was in there, was black. It just looked perfectly burnt, really. In April 2016, so a year after treatments, the surface of the control and the bog water plots has risen, both externally and internally. And it's got pretty much the same vegetation composition as previous. Whereas in the seawater plots, there's a very flat topography within there. There is some evidence of regeneration of sphagnum. There are a few green shoots. It's quite hard to kill sphagnum. And the Eriophrym was starting to regrow as well. And then finally, July this year, the plots are disappearing in the control and the bog water, but the seawater is still very evidently quite badly damaged in terms of the vegetation, but we're getting more sphagnum growth coming back. So I'm going to go through the gas fluxes now for and comparing July 15 and July 16, sort of representative for the summer periods. And I'll keep the pictures up here for those two just to compare as we go along. I'm going to show the bog water values here as well. Error bars are one standard deviation, and all the values are in grams of CO2 per metre square per hour. So in terms of respiration, so this is the CO2 leaving these plots. It's been reduced in both the seawater and the bog water treatment. So regardless of the flooding, there is an effect there immediately after in the short term compared to the control. In the following year, it separated out a little bit there. So we've got the seawater, which is more strongly affected than the bog water than the control, although the values overall are a bit lower for the control as well. So there's some interannual variability in bogs as well to take account of. Gross primary productivity. So this is the carbon dioxide being sucked out of the atmosphere and into the bog. And we want to see negative values on here, which means that it's potentially sequestering carbon. So again, seawater, there's quite a pronounced effect there with, in both years, much lower. In the first year, we've got the bog water, slightly lower than the control perhaps, but not really much of a difference in 2016. Again, maybe this was something of a high value in 2015, but it seems like there's a fairly clear effect of the salt water. Net ecosystem exchange is the combined respiration and the primary productivity. And all the plots are still functioning as sinks. They're all taking the CO2 from the atmosphere, less so for the salt plots. This is really driven by the gross primary productivity. So the methane I've converted to CO2 equivalent, so we're using the same scale here. And methane is quite a strong effect there of the seawater, bog water's not really having much of an effect, and these are very wet places. The water table's often only a few centimetres at most below the surface. So a pronounced effect there in 2015, and it's still there in 2016. And so the combined effect of this converted methane value with a net ecosystem exchange, although in 15 we've got an effect of the bog water flooding, yeah, I don't really know about this actually at the moment. So the working is sync still. It's still operating. I think there's some sort of interannual variability going on here. I sort of expected a stronger effect of the seawater overall. That hasn't come out so far in the analysis, but that's perhaps something else to look at later. So conclusions, a summary of the work so far. So the flooding, regardless of the type of treatment, appears to reduce respiration and the gross primary productivity. But there is something of a lag effect of the seawater later on in the following year. Seawater reduces the methane emissions. This has been seen in other habitats as well, so freshwater marshes, and also in peat slurries, so looking at potential methanogenesis. That's shown that there's competition with sulfate-reducing bacteria. So that reduction was quite expected, really. Overall, though, the fluxes seem to be quite similar. A year later, maybe in a few years, they will come together, or we'll just have more of this variability and we'll be able to see a separation in the responses of the treatments. The big effect, really, was with the vegetation. That's a massive effect there on the sphagnum. It was looking incredibly unhappy. It is able to recover from this sort of one-off treatment. The plots were probably too small to really capture any type of community change, but that's, again, something for future work. And that's where I'm up to so far. Thank you very much.
Great, thanks very much, Martin. We have time for a couple of questions. Any questions? Oh, loads of hands suddenly. Over in the aisle there.
No, you can go on, sorry. I'll think about my questions.
I don't mind who. Go on, Francesca.
That's a really interesting talk. I was just wondering whether you've had a chance to look at any below ground properties.
No, I haven't. No, sadly not. There's so many opportunities really for different angles for this. No, I haven't yet. Were you thinking microbial? Yeah. I think that's the big question really at the end of all this work. What's happening down in that black box? I think somebody called the presentation. So yeah, future work is going to be an exciting field. I think.
Okay, down there then.
Yeah. My first measurements were the week after, once the water table had dropped down to below the sphagnum surface. I started taking some early measurements. I didn't show it in this because probably I'm not very comfortable with it at the moment, but there was a big spike in methane production and respiration following the treatment a week later. So it shot up and then everything died back down again and stayed calm for sort of the April, May period. And then into June, everything, when productivity is increasing, it all started to change. Yeah.
So I guess what I'm asking is those differences you had similar moisture between the neutral and the bogs. There's no diffusional limitations, for example.
Yeah. Those values were sort of representative for the June, July, August period each year as well. But there is a bigger story behind this. It's just yet to come out.
Now, we're running a little bit short on time here. There were, I think, 2 other questions, if you can make them very quick. At the top there first. That's you, yeah.
First, very good to see you using Cosmopnova research. That's good. I just wondered how much you think that coming back to the below ground circumstances, because under Cosmopnova there is quite a lens of brackish water, and how much you think this site has already got some tolerance to seawater as a result of that, which you may not get in some other places.
I don't know. Recently I've been previously about storms and the and salt loading in this area and near Aberystwyth. So yeah, maybe the sphagnum as well, the surface vegetation. It'd be nice to compare some populations. And yeah, within the peaks as well. Hydraulic conductivity is rather poor within peat. The lower down you get, I don't know where that brackish lens. I've spoken with the manager there, but I'm not entirely sure where it is. I don't think it affects the area I'm in.
Okay, we are going to have to move on, I'm afraid, but thanks very much, Martin. So, our last speaker this morning then is Bjorn Robruck on Do Peatlands Hibernate? So, again, wonderful title, over to you.
So yeah, thank you. Yeah, do peatlands hibernate? Well, when Alan Dorrepaal visited our lab in 2013 as a visiting professor, we wanted to make use of an experiment which Luca Bragazza and I started a few years earlier on how peatlands are affected by changing environmental conditions, and especially winter conditions in this case. And then in our lab there was also Robert Mills, who's also in the room here, who was actually one of the driving factors and one of the driving people behind this research as well, joining me every week to shovel away lots of the snow, which was not insignificant, as I can tell you. Climate change is a very important issue nowadays, and it's not only recognized by us scientists, but it's also a concern in community. Like if you open newspapers, you can see these kind of statements like, "It is cold and my car is buried under snow. Is global warming really happening?" So there is a concern. Recent winter warmings may even be cooling climate change concern. Like if you see President-elect Donald Trump even saying something about that it should be warmer because it was so cold when he was walking in Washington. So there is a concern. As scientists, we know that climate change is happening, and most of the time when we've seen some presentations here, we are thinking about drought and increasing temperatures. But there is also concern of decreasing snowfall and decreasing precipitations in winter, and that can seriously affect how ecosystems are working. And we wanted to figure out how this affects peatland ecosystems. So here we can see that this is a paper from the Journal of Climate, that in the northern hemisphere we get a decrease in winter precipitation, so basically snow. And as you've seen from Andre's talk, if you increase or if you decrease the amount of snow, you basically take away a nice warm blanket from an ecosystem, which is then prone to below zero air temperatures, and you get soil frost. And as Andre has shown you for Plantago lanceolata, that can have effects on plants and also soil and microbial processes. So northern peatlands are very important in our climate because they sequester lots of carbon. Martin just told you how the principles work. So the plant community takes up carbon dioxide, and some of that is produced is going into the organic matter part as peat. Some is also leached as rhizodeposits. That feeds the microbial community, so that can feed back then to methane emissions or carbon dioxide emissions, etc., etc. So we basically know, well, we know a lot about how these processes work. But how do they actually work if we have a nice warm blanket of snow on it? And even what are the effects or if we remove this kind of nice warm blanket of snow and have these ecosystems prone to freezing? How does that affect, for example, below ground activity? And we were measuring root activity by nitrogen uptake. And what would that? How would the potential activity of, for example, the photosynthetic apparatus of species, how would that be affected? So winter is very rich in processes. We know, like the cold season emissions, as this paper shows, is very important and dominates the methane budget in Arctic systems. But we don't know much about more temperate peatlands, more temperate peat systems. But so we set out to research this. So we wanted to answer the questions like how do above ground and below ground processes affect nitrogen and carbon cycling, for example, and how does that play out in winter? And we were expecting like light-dependent processes, like photosynthetic capacity, and also nutrient allocation to the shoots, that would be very heavily affected by winter conditions. And that light-independent processes like microbial activity or plant nutrient uptake would be much less affected. And also we wanted to know how will the removal of snow, so the increase actually in frost, how would that also affect that sensitivity to winter conditions? So we set out to the Jura Mountains in Switzerland, where we have a nice little peatland called Praraudet. And this is Constant, one of the ecophysiologists in our lab, who set out with this nice fancy equipment to measure photosynthetic activity. And we did that, we started to do that in winter. And we had this nice 1.5 meter blanket of snow on it, which we needed to shovel down. And we needed to get this kind of system where we had a control situation, and we replicated this all 5 times. Nice control, and then we shoveled away all the snow to get to the sphagnum layer and the vascular plant layer, and had these systems exposed to air temperature, just to the air. And as you can see here, this is how it looked like. This is the snow depth over the control system, in the control plots over the year. So we had a nice layer of snow going up to 1.20 meters, in some plots even 1.50 meters. And then, well, Rob and we did most of that snow shoveling work. We managed to get away much of the snow. Sometimes we went out there, and then the next day it snowed again, and we needed to go back. Which was really nice, and we were actually quite successful in removing the snow from these systems. And one of the things which we did in a few times during the year, so denoted by N1, N2, N3, N4, we added nitrogen, labeled nitrogen 15N to these systems by just digging on the sides of these. Well, here we could just inject it. And here on the controls, we dug a nice little staircase into the snow. Then from the side of the plots, we dug a tunnel. We injected the 15N. We closed the whole system again. Next week, we went back, opened the whole system again, and took out the sphagnum and the vascular plants which we wanted to measure. So some of the environmental conditions here, you can see daily air temperature over the season. You can see that the minimum sometimes goes up to minus 27 degrees Celsius. And so this is just air temperature. And here you can see the daily soil temperature in the peat. So this was measured 2 centimeters deep in the peat. And I highlight this in the situations where we injected the 15N in the system. And here you can see that when you remove the snow, you get these freeze-thaw cycles in the system. While if you have a nice blanket of snow, you have a non-freezing conditions, almost 1 degrees flat temperature. If you then go a little bit into the situation where the snow is disappearing slowly, also from the control plot, so we going into spring, then you see completely the opposite. Like where you still had a little layer of snow, then you had these still flat temperatures. Here the snow disappeared a little bit. And so the temperatures in the peat also rise, but here where you have no snow, there's actually large fluctuations in temperature. And then later in the season, of course, everything was similar in these systems. So actually, these kind of situations, I want to focus a little bit on, like how did these freeze-thaw cycles actually affect the physiology and the nitrogen uptake of different species in our system. So first, photosynthetic capacity. Like, we measured that on Sphagnum magellanicum, and we measured that on Eriophorum vaginatum. And what we basically did, when we took these species, and then Constant measured under ideal conditions if these species were still able to photosynthesize. So under nice, warm conditions, so we took these out of the peat and put them in the cirrus, and measured basically if these species were still able to photosynthesize, so maximum photosynthesis. And if you see here that if you remove the snow, comparing to the control, so the black is the control, here you see the snow removal. So if you have these freeze-thaw cycles, sphagnum is very, the maximum photosynthetic capacity, so the ability of these species to photosynthesize is decreased. So it's very much affected by removing snow in these systems. And also, Areophorus vaginatum is also quite much affected. But the difference between these species is that Sphagnum magellanicum does not recover very fast. So it is affected by these freeze-thaw conditions, and it does not recover very fast, while the Areophorus vaginatum is also affected by removing snow, but when you move into the growing season, it's actually the difference between the control and the snow removal is not that big anymore. Actually, it disappears. So you are already feeling a little bit that you can change the competitive balance between the sphagnum community, which is so important for the carbon sequestration in these peatlands, and you change the competitive balance with these vascular plant species. And this we also did in a little side experiment with a student from Estonia, a PhD student from Estonia, Martin Kutim. And he also did some freeze-thaw cycles like Andre basically did with his Plantago lanceolata. And here you can also see that if you have sphagnum growing in freeze-thaw conditions, its mean photosynthesis is very much affected compared to the control. And this is probably done by a depletion in the chlorophyll A content in this sphagnum. So you have a reduction in the maximum photosynthesis, which is probably due by a damage in the photosynthetic apparatus. So that has effects on above ground, below ground production. Well, actually not so much on above ground production. This is the vascular plant biomass, and if we harvested that in all our plots, so these different bars are winter, early spring, late spring, summer, and you can see that there is not a very big effect of control and snow removal on the above ground production. But then below ground, we can see that if we took below ground, so root biomass, then if we remove snow, that really had a large effect on the amount of roots which were produced in these systems. And also the biomass production of sphagnum was really highly affected. So above ground biomass not affected, and snow removal reduced below ground biomass in winter. But there was a fast recovery again into the spring shoulder season and into summer. And peat moss is strongly impacted. So now the effects, and I need to hurry up a little bit, the effects of 15N uptake. And if you look at these lower graphs, which is the 15N recovery, so this is basically the amount of nitrogen 15 which the plants could take up. You can see here above ground and here below ground. Above ground, so in the plant tissue, we could not find much differences between the control and the snow removal. So there was not a difference in the amount of 15N which we could trace back in the plant tissue. But if we were looking at the root concentrations of 15N, we could clearly see that if you had a nice snow pack on it, so these nice flat temperatures not below 0, so 1 degrees Celsius, that we had a nice uptake of 15N. So in these systems they are actually taking up nitrogen if there is a snow cover. And if you remove that snow cover, there was a huge decrease in the amount of 15N which could be taken up by these plants. So to conclude, peatlands are active in winter and especially below ground. Few processes actually shut down. And then a continuous snow cover is actually crucial for many of the processes we measured in winter, and especially for below ground processes. And so these people I need to thank. And just a little advertisements. I'm moving to the University of Southampton, and I have a PhD position available. And if you're interested in that, just have a look at my website. Thanks very much.
As you're the last talk, I'm going to allow one question if it's quick. before you go, I'm going to invite you to thank all the speakers in a minute. Before you do that, though, a quick advert. The BES has a climate change ecology special interest group, which I chair. It'd be great if we had everybody here sign up to join us. It's only a commitment to join a mailing list, essentially, and then we organise various meetings. Send an email to climateChange@BritishEcologicalSociety.org. And that's the end of the session, but let's just thank all of the speakers. It's been a really good, really varied morning, and all of the presentations have been fantastic. Thank you.